Oh, okay. All right. Hello, everyone. Welcome to another episode of Let's Discuss the Paper Right After Reading It. I'm the host, Qian Chen, for today. And in this kind of episodes, we read the paper within 2 hours and then come back to discuss it. So today we are reading a paper by King and colleague, published on Journal of Neuroscience. Yes, Neuroscience in 2021, titled "The Human Brain Encodes a Chronicle of Visual Events at Each Instant of Time Through the Multiplexing of Traveling Waves." And let's introduce ourselves first. My name's Qian Liang, and I'm from Monash University, and I'm a PhD student.
Yeah, I'm Aniko Kustar. I'm also from Monash University, and I'm also a PhD student.
I'm Yota Kawashima. I'm a research assistant at ATR Japan and Monash University.
Okay, so I will start by summarizing this paper first, and then we will go into a free-flowing discussion phase. So for this paper, the background is that there have been numerous research attempts to kind of investigate the processing of visual inputs, but most of them are done with presenting one static image to the participant, and you see how the information goes from low level to high level in the brain and processed in an increasingly complex manner. But and what they found was that this kind of visual input goes from the low level early visual cortices, and then it travels through the ventral and dorsal pathways of the brain, in which the visual features they captures go from very simple like luminance, spatial frequency, to something more complex and meaningful and abstract, such as the presence of a face. This whole process, what they found was that typically takes around 1 second to complete. And this leads to a question of if it takes 1 second for the brain to process image in full, then how do we manage to see a sequence of image, each of them presented for way less than 1 second? How do we manage to not mix up the features from one image to another? And this is the focus of the current paper. To address this question, they did the EEG combined with behavioral paradigm kind of study. For the behavioral part, they have the participant to view a successive sequence of 8 Gabor patches in varying orientations, and each patch was presented for 250 milliseconds. And to keep them attended to the task, they asked participants to do some very simple decision task and just button press. And in each trial, they see a sequence of 8 stimuli, and they had the each participant to do 600 trials or something, and which cumulates to 5,000 Gabor patches viewed over the whole experiment. And while they're doing the behavioral tasks, they record participants' EEG signals. And with this paradigm, they did their analysis. There are 4 main analyses they did. So the first one is they try to see whether EEG signal correlates with the orientation of the Gabor patches. And the second, they want to see whether the brain tracks the changes in the orientations of the Gabor patches real time. And the third analysis is to further investigate whereabouts in the brain that kind of does the real-time tracking job. And finally, they model the neurodynamics of the brain during this whole process to see how exactly the brain regions talk to each other to complete this change tracking task. So what they found was that first, brain signal, EEG signal, does track the angle of each stimulus real time. But it's not just the EEG signal you get right after presenting one stimulus. So it's not just the signal after presenting one stimulus that correlates with that stimulus. Instead, you can actually decode the angle of one stimulus from the brain signals that's 5 stimuli after. So in a sense, your brain can host up to 5 stimuli about the orientations at one time. And similar to, this is the similar situation with how the brain tracks the change in stimulus orientations. What they found was that the brain signal can track the orientation change in stimulus in a sequence of up to 5 stimuli. And next is the question of whereabouts in the brain that does that, record these angles and angle changes. What they found was quite similar to these static image stimuli, in which the coding of orientation and the change of them first emerge from early visual cortices and then spread to inferotemporal and dorsoparietal cortices. And further, the coding of the orientation change would spread to the superior frontal lobe as well. Finally, how does the brain regions talk to each other to track this, to track the orientation? So what they found was that, I don't fully understand this part actually, but what they found was that each time the participant sees a new stimuli, the new visual input comes in, it would ignite a small kind of inhibitory signal from the higher level to kind of update what's happening in real time. So to summarize, what this paper found was the brain tracks and updates visual input over a sequence of stimuli. And such ability is hosted by a long chain of areas from early visual cortices to via the ventral and dorsal pathway, and some even goes towards the frontal area. That's it. And now let's move on to the discussion section. Anyone has anything they want to mention? Or anything to add as well?
I guess it was, for me, somewhat unfamiliar concept, but then I guess it has been researched quite extensively, this concept of traveling waves, which seem to be like, I guess it is mostly in the gamma frequency range, so like above 40. Am I saying that? No, that's not above 20 hertz. Oh wow, I'm having a brain freeze now. Anyhow, so like a very high frequency, like an activity in a very high frequency range, and I know that has been mostly connected to visual feed-forward processing. And yeah, I think it's an interesting concept, and it's an interesting way to examine the information flow in the visual cortex. but I haven't really heard of them a lot actually. So I was interested to read more about them.
Okay, so I feel, first of all, this study is really interesting and kind of makes sense because, we know that the visual processes is processed from, back to the forward, and that can be kind of tracked in terms of easy recording. That's really great. And also, I'm kind of interested in like how long will it take for each region to do their computations, and this study kind of show that then within like a 1 or 2 seconds, these processes can be done. So yeah, it's really interesting paper. If I ask one question, or I also interested in, in the discussion section, they mentioned that limitation of this study, saying that, here they use the artificial stimuli, but it's really interesting if they use or do the same thing for the natural, natural scene image, because I read that low spatial frequency information will be, processed or propagate to the frontal area quicker than the high spatial resolution information, so that the brain can, first kind of make a guess about the context or the background of the image seen. And then after that, by using that, the brain can start to recognize the object in the scene or things like that, which require the, high spatial resolution, high spatial frequency image. So yeah, the same approach for the natural scene image will be very interesting.
Yes, and you might see some more distinct neurodynamics for natural images, because I also read somewhere that proposed that in order to recognize a specific object in a scene, you need recurrent processing. So if you do it on natural images and you analyze this neurodynamics, you might be able to see kind of recurrent connections.
Yeah, and I guess to that, I think the this, like they again also mentioned this in the limitations, but the fact that this is not meaningful stimuli, or not necessarily, and not expected stimuli, may be actually so the fact that they have found this kind of 1 second time limit and like 5 stimuli maintenance, it very well may be that this is because of the exact experimental conditions and stimuli. And potentially, yeah, I know that we have read a paper a few months ago where they looked at, I think, naturalistic images, and they were able to record our objects. Anyhow, so I think they used prefrontal regions to decode, and again, it was quite a long time, quite an extended period where they were able to like decode the specific image that was presented. So yeah, I think perhaps if you have more kind of naturalistic images and other cues and other like components of the image that can be stored and connected to different things, it might be even like potentially longer or more image can be maintained in in the visual processing system.
Yeah, and regarding that, actually, I also have very similar questions. So the entire experiment setup is kind of tune the participants to focus on the orientation of the patches. And that might have that might have made the processing more efficient in a way, and probably make the change tracking kind of signal more obvious, maybe. Yeah, it's neuroscientific studies are not my strong suit, but it could be a guess. So with unexpected stimuli or meaningful stimuli, it could actually go both ways in terms of whether the brain's better at tracking them or worse at tracking them. Because on one hand, you have more meaningful contents for you to tag, but on the other, there are many visual features in the scene, and it's quite unexpected which one stands out next in the next image. And you don't have one single feature for you to track all the time. Maybe color, but it's hard to say. Oh, I think there are studies that talk about the spatial organization of the scene is very critical for quick scene understanding. So maybe the brain could utilize that kind of information to host more stimulus at the same time, compared to artificial stimulus that are fixed in frequency.
I think the paper you mentioned is the one we read during this journal club. I think the review paper of like a spatial attention something. Yeah, I remember like maybe 3 weeks ago or something.
That one is about like how objects and things kind of organize meaningfully in a scene. What I read was something earlier than that. I think it's called spatial envelope hypothesis or theory.
Okay. I see. But anyway, I have another, not the question, but the comment that's about the task for participant. Here, participant need to pay attention to the angle because the author of this study want to know whether participant pay attention to the stimuli or not. But in that sense, the participant were consciously perceive this stimuli, right? So I was wondering whether we can do something similar in terms of, let's say, dual task framework or not, and then maybe for unattended stimuli, we will see the different, the traveling wave maybe stop at the parietal region and does not go to the frontal. And then we can say, oh, for conscious perception, we don't need the, frontal region or something like that. So in that sense, yeah, this study is really, interesting and useful for further, investigation.
Yes, I was thinking, was this orientation differentiation the attentionally demanding task?
I think it is boring as hell. And so it is like not attentional demanding, but you need sustained attention to like just make yourself listen or like pay attention to this boring thing for thousands of trials.
Oh yeah.
But like, yeah, I think that was more in a way like, so yeah, so we like whether attentionally demanding, it wasn't demanding, just by itself, but it just, I think along the task, I reckon it must have been demanding on the participants.
Oh, yeah, true. Very true. 5,000 patches. Actually, I was thinking about maybe they can try, because we bring up expectation. It's possible to make the orientation change of the patches either predictable or unpredictable. And I was reminded that this has basically already been done. So if what they found was if the trajectory of an artificial object is kind of predictable, let's say you just go like this, then compared to when it is unpredictable, I think like you can decode the location of that object in the brain earlier when it is predictable than when it is unpredictable. So yeah, the brain takes extra time to kind of adjust for the kind of change. And I think that's pretty interesting as well.
Yeah, definitely. I have a more kind of consciousness related or more kind of zoomed out question, which is, what do you think, how should we, or should a theory of consciousness be able to explain this kind of maintenance of different stimuli in the visual system or not? Because I'm not sure. So in this paper, for example, they have bring up, I think, the predictive coding and recurrent processing, which are definitely, I think, important candidates in and can explain some aspects of conscious processing. But yeah, I was just wondering if a consciousness theory doesn't able to kind of account for this maintenance of or kind of decodability of images presented long ago and maybe not be in the focus of the attention or yeah, not being actually yeah, I don't know. So like potentially, yeah, I mean, so basically I don't think that anyone doing these experiments actually consciously experiences the 5 Gabor patch before, but just the one that they are actually looking at or how they are changing it. So
oh, I see your point.
Yeah,
it's like they probably don't remember what the patch was five patches before, but it's actually evident in the brain, right? Is that what you're suggesting? But
yeah.
Well, I mean, so long as they consciously experience that one, the one they've seen when they see it, then I think it's fine.
Or I mean, I guess they were counting somehow the orientation, so they will have some like representation, I guess, of what happened, but maybe not like a visual. So like it, I don't know. I guess we don't know how people think or how people solve this task, but I would imagine, or if I imagine solving this or doing this task, I would be like kind of counting or trying to remember, like, okay, this is diagonal, this is on like what was the other? So like basically the orientation. So I'm able to say that, okay, most of them were diagonal, but wasn't attempting to visualize all the previously presented Gabor patches and once est count them at that moment, if that makes sense.
Are you talking about something similar to like the strength of incidental memory?
Yeah, well, like I'm just wondering whether this kind of like, I think it's very useful to know that at least in the visual system you have specific traces of these stimuli the kind of available or decodable, but then how is that actually influences what you experience now? That's my question.
I suppose people can feel because people can report the general direction of the entire sequence, even though it's possible that they don't remember a particular one from the sequence. I think that actually corresponds to how the change signal could spread to the frontal, but not the rest of them.
Yeah. I think, yeah, anyhow, I was just wondering about this.
And I know that I don't know how to pronounce his name, but there used to be this paper that now was collaborating in that looks at the strength of incidental memory on faces. And in this study, they asked people to look for a particular face in the entire crowd. And what they test was the other face that people scanned on the way to that target face. And in principle, you're not supposed to remember those irrelevant faces well, but they actually found above chance performance in recognition with above chance conscious access. So, you know, I think that might kind of answer your question, but then again, that was faces. They were faces and they were easy to remember for humans. Possibly not the case for something as abstract as Gerber patches.
Yeah, that's a good point.
Yeah, and I think another thing is that if we can measure the delay between the stimulus onset and the conscious perception, and then see, let's say we found the delay of 50 milliseconds, and then we can predict that then, the region that, correlate, or the brain region that generate conscious perception should be encode that information at that time, like a 50 millisecond. And let's say this, paper, they show the, gradient from the back to frontal, and then we can, predict, okay, this region is important for conscious perception. And that's another thing we can do to, relate this study with conscious, experiences, or the theory of consciousness.
Any other comments?
I'm just thinking about this, what you said, Jutta, because I think this has been done before. Like, of course, this attempt has been done on, like, okay, looking at what happens or which regions are active after 50 milliseconds or so. And I mean, I think that was this, maybe I'm probably wrong, but I'm wondering if the P300, this might like a more later component, but I know that has been suggested as a potential EG marker for conscious update of the working memory, and then people were looking at P300 as a potential marker of conscious awareness. But then, of course, there are also earlier potential ERPs. So yeah, I think it's difficult, but I'm sure that this has been done in one way or another. But I can't recall a concrete study actually.
Yeah, it was probably not possible in the context of this study, because I think the each time window is like around 250 milliseconds. So no time for P300, I guess. Well, they can still look for the P300 of the previous stimulus after.
Yeah, so there's actually some techniques to kind of decorrelate the other like activity related to the previous stimuli, because you can just model that, okay, at each time point we have an expectation of some slope, and then you say that, okay, we have these expectations after each other, and then just extract it from the next one. But yeah, otherwise I don't have any more questions.
Okay, none from me.
Yeah, another thing is that maybe this study can be useful for the masking, literature, because we can say that, maybe in order to explain the backward masking phenomena, by psychophysics experiment, we can, or we have some threshold for backward masking, the delay threshold. Then by, that knowledge, we can, expect, or in combination of this study, we can say that, or in order to achieve the masking, we need this region, I mean, this brain region and this brain region, and then we can, investigate the mechanism of, masking, maybe.
Oh, and also you can kind of test the strength of the masking. You see whether the trace of, the initial stimulus is still present after masking. I know that there have been studies that already look at, how masking disrupts the recurrent processing and therefore disrupt perception. But yeah, I think this method would be an interesting way to systematically quantify the effect of masking.
But I was thinking, so because it seems that time is an important kind of variable here. So potentially the strength of the masking would be strongly related to the length of how long the masking is presented.
Yeah, exactly. Just if like 5 is not enough, we just add like 3 more.
Yeah. Yeah, no, that would be very interesting, actually.
Okay, any other comments? No? All right, then this concludes today's episode. See you next time.